imagine, just for a second, if your nose wasn't just this simple passive tunnel for air.
Right.
Like, imagine instead that it's this incredibly precise, three-dimensional chemical sorting center, and it comes with a built-in GPS.
Yeah, a literal coordinate system.
Exactly. Think about your own nose right now. Every single time you breathe in the smell of, fresh coffee or rain on hot pavement, specific, exact coordinates in your brain are lighting up with just pinpoint accuracy.
It really reframes how we tend to view our senses. Because we usually experience smell as this sort of vague, atmospheric, almost invisible thing.
Yeah, totally.
But neurologically speaking, it is an incredibly highly structured, deeply physical process.
And today, our mission for this deep dive is to explore exactly how structured it really is. We're unpacking a truly groundbreaking 2026 paper
From the journal Cell.
Right, published in Cell by Bintu and colleagues. The goal here is to exhaustively map out exactly how the mouse primary olfactory system detects and spatially organizes social and predator odors. And we are going to leave no stone unturned here.
No, we're diving deep into all of it.
Every single mechanism, every spatial gradient, every molecular name tag, we are decoding the entire source material.
And, while humans are obviously highly visual creatures, taking the time to understand this molecular atlas in mice, it actually unlocks the secret to innate mammalian behaviors across the board.
Right, because it's universal hardware.
Exactly. We're looking at the fundamental hard-coded wiring of survival. Like, this research explains the mechanics of how a mother instantly recognizes her specific pup, or how a mouse just inherently knows the cat is nearby purely through a stereotyped chemical map.
The literal hardware of instinct.
Yeah, beautifully put. It's the hardware of instinct.
So, okay, if we're going to map out this entire olfactory system, where exactly are we starting? Like, what is the physical territory we're looking at when a scent actually enters the body?
Well, it starts the moment a mouse breathes in. The airborne scent molecules travel up the nasal cavity and they hit a specialized tissue.
The MOE, right?
Right, the main olfactory epithelium, or the MOE. You can sort of picture this as the delicate lining inside the upper nose. And embedded in this tissue are millions of olfactory sensory neurons, the OSNs.
And they have these tiny little hairs, don't they?
Yeah, cilia. These neurons have tiny hair-like structures that literally reach out into the mucus to, physically catch the odor molecules as they pass by.
Okay, so the odor molecule physically binds to the neuron.
Yes, and here is the absolute defining rule of this entire system. Each one of these individual sensory neurons expresses exactly one olfactory receptor gene, just one.
Out of a massive family, right?
Right, one OR out of a family of about 1,000 different OR. So once they catch a scent, these neurons generate an electrical signal and send it down long biological wires.
Or axons.
Exactly, they're axons. And they send that signal straight back into the brain, specifically to a structure called the olfactory bulb.
Wait, hold on, though. Let's unpack this for a second. If you've got 1,000 different types of receptors, and they're all just mashed together across millions of cells in the nose, how on earth are the researchers isolating them without just getting biological static?
It's a huge problem.
Right. Like, how do you map 1,000 overlapping data points without getting hopelessly lost?
Well, that specific problem actually held the field back for decades. The solution that Bintu and the team used is this incredible piece of imaging technology called MERFISH.
MERFISH? Sounds like a cartoon character.
It really does. But it stands for Multiplexed Error-Robust Fluorescent In Situ Hybridization.
Okay, that is quite the mouthful. Let's try to visualize this. It sounds like trying to map a massive stadium filled with millions of fans, right?
Yeah.
And you're trying to find 1,000 specific groups of people in that crowd.
That's a perfect analogy, actually.
Yeah. Because instead of taking a blurry aerial photo where everyone just kind of blends together, MERFISH is basically attaching a highly specific glowing barcode to every single person's shirt. So suddenly, you can look at the stadium and see exactly where everyone is sitting, all at the exact same time.
Yes. But not only does that glowing barcode tell us who they are, the researchers had to be incredibly clever about where they attached it on the molecule.
Oh, why is that?
Well, because they are looking at RNA, the molecular instructions the cell uses to build these receptors. And they ran into a major hurdle. The 1,000 olfactory receptors are genetically very, very similar to each other.
Oh, because they're all basically doing the same job, right?
If the researchers designed their probes to target the main coding sequence of the genes, the probes would just accidentally bind to the wrong targets. It would be, as you said earlier, entirely biological static.
Oh, I see. So they couldn't target the main body of the gene because they all look too much alike. What did they target instead?
They designed their MERFISH probes against the untranslated regions, or the UTRs.
The UT.
Yeah, these are the specific sections at the very ends of the RNA molecules. Think of them like bookends. Even if the protein coding parts of two different receptors are, say, 90% identical, their UTRs on the edges act like highly unique molecular fingerprints.
Wow.
And it gets better. They also used what are called split probes.
Wait, what's a split probe? Like they cut the barcode in half?
Basically, yeah. A visual signal only lights up if two separate distinct halves of the probe find their specific target and bind right next to each other on the RNA.
Oh, that's brilliant. So it effectively eliminates false positives. If only one half binds, you see nothing.
Exactly. And since RNA inside a single cell can be incredibly scarce, it's really hard for a microscope to even see that tiny glowing tag.
I was going to ask about that. How do you even see it?
They solve that using multiplexed branched DNA amplification.
Branch DNA.
Yeah, because RNA is so tiny, they couldn't just use one glowing tag. They
No.
literally coaxed the DNA to grow microscopic branching trees on top of the target.
You're kidding.
No. These branches provided hundreds of docking stations for fluorescent molecules to latch onto, artificially boosting the glowing signal until the microscopes could clearly read it.
That is just wild. I mean, they are engineering glowing microscopic DNA trees on the unique edges of RNA molecules just to map a sensory organ.
Science is amazing, isn't it?
It really is. And the result is that they successfully mapped the near-complete repertoire of about 1,100 ORs. And the source mentions they also mapped another specialized group called TARs, about 10 of them. What exactly are TARs?
TAR stands for trace amine-associated receptors. Trace amines are essentially the chemical byproducts of amino acid degradation.
Okay, so like breaking down proteins.
Yeah. In the real world, you find them heavily in the sweat, urine, and bodily fluids of other animals.
I see where this is going.
Right. So while your standard olfactory receptors are smelling general environmental odors like coffee or rain, TARs are highly specialized evolutionary alarm bells. They are tuned specifically to the presence of other living creatures, whether that's a potential mate or a deadly predator.
Okay, let's do a quick mini summary here for the listeners, just to make sure we're keeping the big picture in focus. So the MOE, the tissue in the nose, catches the smell. The olfactory bulb in the brain processes it. And this MERFISH technology acts as the ultimate glowing barcode scanner, reading the exact layout of over 1,100 distinct receptors.
Including those trace amine alarms, yes.
Right. Which brings up a fun thought experiment for you listening at home. If you could attach a glowing barcode to any cell in your own body to see exactly what it's doing right now, what would you choose? Think about that.
Now that we have this scanner, the next logical question is what the map actually looks like.
Right, the actual layout.
And it turns out the nose is not a random chaotic soup of cells. It is highly organized along two specific spatial gradients.
Okay, the GPS coordinates of the nose, how are they laid out?
Gradient 1 is central to peripheral. If you imagine looking straight down the nasal cavity, it's like a bullseye, right? Or concentric rings expanding outward from the center to the outer edges.
Got it, bullseye.
Then gradient 2 is apical-to-basal. This refers to the depth or the layers of the tissue itself. The apical layer is the top surface right at the lumen, where the inhaled air physically flows. The basal layer is deep down at the basal lamina, the foundation of the tissue.
So every single sensory neuron has a specific hard-coded address on these two axes.
Every single one.
That is literally a biological X and Y axis. But, okay, if a cell is sitting at a specific coordinate in the nose, where does its wire go in the brain?
Well, this is where the structural elegance of the brain really shines. The spatial gradients in the nose perfectly mirror the spatial axes of the olfactory bulb.
Wait, I actually have a great memory trick for you, for the listeners, so you can visualize this mapping. Just remember, CD, AA.
Oh, I like that.
Yeah. Central to dorsal, apical to anterior, CDAA.
That perfectly encapsulates it. If a neuron is located in the central part of the nose, its axon travels to the dorsal, or the top part of the olfactory bulb. That's the CD. And if a neuron sits in the apical, or top layer of the nose tissue, it projects its wire to the anterior, or the front part of the bulb. That's the AA.
And the microscopic precision of this wiring is just staggering to me. These long projections travel all the way from the nose into the brain, and they land at their target within 200 to 300 micrometers of accuracy.
Which is incredibly tiny.
Yeah, to put that in perspective, that is the equivalent of just 2 or 3 tiny biological junction boxes, which are called glomeruli.
Right. A glomerulus is basically a spherical cluster where the wires from the nose synapse or connect with the neurons of the brain.
And when the researchers compared the map in the left brain hemisphere to the right brain hemisphere, it was nearly 99% identical, a virtually flawless mirror image.
But this raises a fundamental biological question. Axons are just long biological wires growing through tissue. They don't have eyes.
Right.
So how do they navigate a three-dimensional space to find a specific target that's only 200 micrometers wide?
Yeah. How does the wire know its own CD, AA coordinates?
To figure this out, the researchers had to uncover the algorithm of the brain. They integrated single-cell RNA sequencing to look at what else these neurons were manufacturing besides just the receptors.
Okay.
And they discovered that neurons destined for specific regions of the brain express specific guidance molecules. These are genes like NRP1 and Plexana1.
So they're essentially building their own navigation systems.
Exactly. They also found specific G protein and CAMP signaling pathways. As the tip of the axon, which is called the growth cone, pushes through the tissue, these molecules act as biochemical traffic lights.
Traffic lights, I love that.
Yeah, they literally read the chemical gradients in the surrounding tissue, actively steering the growing wire, telling it, turn left here, follow this chemical trail, stop growing right here.
Okay, let's pull all this together for our 5-minute recap. We've covered a lot.
We have.
Bullet 1, 1 neuron, 1 receptor. Bullet 2. We've got the physical hardware of the nose and the brain, and the MERFISH scanner barcoding 1,100 distinct receptors. Bullet 3. We have a highly precise map utilizing a central-to-dorsal, apical-to-anterior coordinate system. And bullet 4. We have the molecular traffic lights guiding the whole thing.
The map of the city.
Right, the map of the city. But having a map is one thing. Seeing the traffic actually move through the streets is another. How do we catch these neurons in the act? Like, how do you see a smell happening in real time?
To capture that dynamic function, the researchers utilized something called immediate early genes, or IEGs. Specifically, they looked at a gene called egre1.
Egre1.
Yeah. You can think of egre1 as the molecular equivalent of a cellular check engine light.
Okay, but why an immediate early gene specifically? Why does it have to be immediate?
Because of its speed. When a sensory neuron detects its specific odor, it fires an action potential. This causes a massive influx of calcium into the cell.
Right, the electrical signal.
Immediate early genes like egre1 don't require the cell to synthesize new proteins to turn on. They react directly and immediately to that calcium influx. The absolute millisecond the neuron fires, the transcription of egre1 spikes dramatically inside the nucleus.
Oh, that makes so much sense. So if MERFISH tells us who the neuron is and where it lives, the egre1 gene tells us if it is currently yelling.
Yes. But in a study this groundbreaking, you have to validate your tools. The researchers didn't just assume this overlap would work. They ran a rigorous test case.
Using a chemical called acetophenone. Right?
Exactly. Acetophenone has a distinct sweet floral smell. They exposed male mice to this scent for exactly 30 minutes. That specific time frame gives the egre1 gene enough time to fully spike and accumulate so the microscope can actually see it.
And if I recall from the source material, they didn't just test one amount. They did a dose dependency test, right?
It did. They tested a 100% pure concentration and a highly diluted 1% concentration.
And what happened?
The results were incredibly validating. First, the specific receptors that lit up with the egre1 check engine light perfectly matched the known responses from older, much slower, low-tech studies. It proved the tool worked flawlessly.
Awesome.
Second, the 100% concentration lit up significantly more neurons than the 1% concentration.
So it proves the system captures dynamic range. It doesn't just show on or off. It physically shows that a stronger smell recruits a wider network of receptors.
Yes, exactly.
That's amazing. By overlapping the MERFISH receptor map with the egre1 activity map, scientists can literally sit back and watch the nose thinking in real time.
It's a window into the brain.
Which prompts a question for you listening. Think about the smell of your absolute favorite food right now. Based on what we just learned, What complex combination of your 1,000 receptors do you think is transcribing each one and lighting up your brain at this exact second?
It's an incredible process to visualize. And once the researchers knew they had a validated high-throughput pipeline, they moved away from simple sweet smells like acetophenone and moved into the real world.
Right, getting out of the test tube.
Exactly. This is where we get into the ethological map. Ethological meaning the natural behavioral cues that are absolutely essential for an animal's survival.
And this is where the data tells a truly wild story. They exposed these mice to 4 vital cues: male mice, female mice, pups, and cat bedding.
The ultimate predator scent.
Yeah. So let's look at the numbers. Let's start with the male cue.
The male odor triggered the absolute largest, most selective set of receptors in the entire study.
Yeah.
106 specific ORs.
Wow, 106.
Yeah, and it also heavily activated one of those trace amine receptors we talked about earlier, TAR5. Physically, these 106 receptors are spread very broadly across the entire MOE in the nose.
And where do they project to in the brain?
They project their wires to a large midband and a smaller dorsal region of the olfactory bulb.
Now, 106 receptors seems like a massive footprint for just smelling another male. Why so much hardware?
Well, if you think about the evolutionary pressure on a male mouse, smelling another male is an incredibly complex, high-stakes assessment.
Like turf wars.
Yeah. It's about territory boundaries, assessing social dominance, evaluating threat levels, reproductive competition. The brain dedicates a massive amount of sensory hardware to this because getting the social hierarchy wrong is evolutionarily costly.
That makes perfect sense. Now, what about the female cue?
The neural footprint is much smaller. The female cue triggered only 34 ORs.
Okay, 34 compared to 106.
Right. And what's really interesting is that 60% of those 34 receptors also responded to the male cue.
Oh, wow. So there is significant biological overlap in how they smell their peers.
Exactly. Spatially, these female-detecting neurons are focused heavily in the central part of the MOE and project to the mid-posterior region of the bulb. And instead of TAR5, the female cue triggered a different specialized receptor, TAR3.
Okay, so mates and peers share some underlying circuitry, but definitely have distinct specialized signatures. But what about the cat cue? The predator? I mean, this is pure life or death. How does the brain map a mortal threat?
The predator cue activated neurons located in a highly specific peripheral ring on the outer edges of the main olfactory epithelium.
Okay.
And when you trace those specific wires into the brain, they project to a very distinct, thin middle band located on the extreme anterior and posterior edges of the olfactory bulb.
Wait, a middle band. Doesn't that completely break the old textbook rule?
It absolutely does.
Because for years, the neurobiological consensus was pretty binary. The textbook said that the ventral or bottom part of the olfactory bulb was strictly for attractive smells like food or mates, and the dorsal or top part of the bulb was for aversive smells, the terrifying things like predators or spoiled food. But you're saying the terrifying cat odor didn't go to the top, it went to a middle band.
It completely disrupts the old paradigm. The aversive cat odor bypassed the expected dorsal area entirely.
Yeah.
This singular piece of data forces us to fundamentally rewrite the textbooks on how the mammalian brain physically categorizes fear versus attraction.
That is huge.
It's not as simple as top equals bad, bottom equals good. The spatial domains are far more nuanced. By routing to a middle band, the predator signal might be able to rapidly interface with both pathways, integrating the pure fear response with the motor planning required for immediate escape.
That is just brilliant. Let me quickly synthesize where we are for another quick recap. We've moved from the anatomical structure of the nose to its actual function. We're seeing exactly how the brain uses immediate early genes to flag active neurons, like a check engine light.
Yep.
We saw that male cues activate the most receptors, 106, and we've officially busted the old dorsal means bad, ventral means good rule, thanks to the cat cue data.
Which is a massive shift in the field.
Huge. But we still have one cue left, the pups. And this finding blew my mind. Walk us through how a mother smells her pups differently than a virgin mouse.
So if we look at the bigger picture, it is easy to assume that our senses are fixed. A nose is a nose. Your receptors are your receptors.
Right, like buying a standard radio, you get the channels it comes with.
Exactly. But the data here proves otherwise. When the researchers exposed virgin female mice to the scent of pups, it elicited a very restricted sensory response. Only 18 ORs lit up, along with a specific trace receptor, TAR7B.
Just 18 receptors, that's a tiny fraction of the 1,100 available. But what happened when they tested the mothers, the mice who had just recently given birth?
In the mothers, the hardware literally changed its physical sensitivity.
What?
The physiological experience of motherhood completely altered how the nose reacted. The mothers engaged all 18 of those original virgin receptors, but they also unlocked 11 additional mother-selective ORs that were entirely dark and dormant in the virgins.
So the network expanded from 18 to 29 receptors, just by becoming a mother.
Yes, and the mechanism here is crucial. Not only did they unlock 11 new receptors, but the sensitivity of those receptors was dialed up massively. Those specific mother-selective receptors were tuned approximately 4-fold higher to the pup scent in mothers than in the virgins.
4 times louder.
Exactly.
Yeah.
Spatially, these highly tuned pup receptors live primarily in the central MOE and project their wires to a very small, concentrated dorsal subdomain in the olfactory bulb.
So let me make sure I'm getting this. Going back to my radio analogy, it's like buying a standard radio, but the moment you have a kid, 11 hidden channels automatically unlock, and the volume knob goes up by 400% for anything related to your baby.
That is the perfect way to look at it. The physical odor of the pup in the room didn't change at all. But the internal state of the animal dramatically, physically ramped up the peripheral nervous system's sensitivity to that specific life-saving odor. It proves that sensory perception is not just a bottom-up process driven by the outside environment. It is heavily modulated by the internal state and the life experience of the organism.
The takeaway here is profound. Major life events can literally upgrade your physical sensory hardware. Think about that for a second to everyone listening. Does it change how you view your own senses, knowing that your physiological state can alter the physical sensitivity of your neurons?
It's mind-boggling. And if we synthesize everything we've uncovered today, the sheer achievement of the Bintu et al. Paper is staggering.
Truly.
By combining the MERFISH spatial mapping technology with the EJ1 functional mapping, we finally have a high-resolution, near-complete atlas of how a mammal actually navigates its chemical universe. We aren't guessing with blurry photos anymore.
We know the exact coordinates on the gradients, we know the precise names of the receptors, and we understand the distinct neural domains in the brain responsible for socializing, mating, and surviving. And, while human noses are anatomically different from mice, and we rely much more on vision, the fundamental logic of neurobiology connects us all. We are constantly subconsciously absorbing a vastly complex chemical world, a world that is quietly dictating our moods, triggering our deepest fears, and guiding who we love.
It's all happening right under our noses, literally.
Literally. So we want to leave you today with a final provocative thought to mull over. We saw that maternal experience selectively enhances 11 specific receptors to smell pups 4-fold better. If our profound life experiences and internal states can literally rewire and upgrade the physical sensitivity of our peripheral sensory organs, what other hidden channels have you unlocked in your own senses without even realizing it?
It's a great question.
It really is. Until next time, keep diving deep.